I want to start off talking about what is net filtration pressure. This really has a lot to do with tissue perfusion or bulk fluid movement. If we look at this picture, right, what you're noticing is blood is flowing down the arteriole and into a capillary bed. And you're going to notice at the arterial end of that capillary bed, all of these arrows are pointing out. And really what that is trying to show you is that this is how we are going to take oxygen and nutrients and pull them out of your vessels and into your tissues at the arterial end of the capillary bed. Now you'll notice that as the blood is moving through that capillary bed, it's going from red in color to purple to blue. And that's trying to show you the oxygen's being pulled out. Now here at the venial end of that capillary bed, notice that the arrows have changed direction. And now they're pointing in to those vessels. And so this is trying to show you that this is where we're going to be bringing wastes and CO2 from your tissues back into your vessels. And then from there, that spent blood is going to move out of the capillary bed and into the venial and get returned back to the heart. So really when we talk about net filtration pressure, the focus here is how are the arrows from here to here changing direction? How is it possible? How is it possible to force something out of a vessel at the arterial end, but then force things back in at the venial end? So that's really what net filtration pressure is all about. Okay, so the first thing that you're going to notice, I'm going to do a drawing in just a minute for you, but the first thing you'll notice is that as I go through the drawing, I'm going to talk about two different kinds of pressure at play. The first one is hydrostatic pressure. A lot of times I like to think of this as just blood pressure. All right, BP for short. And so hydrostatic pressure is the force that's inside the vessels and it's pushing things out of the vessel. So that's another reason why I always think of this in terms of blood pressure. It's what's in the vessel. Now the other kind of pressure that I'll talk about is osmotic pressure. This is the pressure that is not in the vessel. This is the pressure that's found in your tissues, so outside the vessels. And this is a pressure that's going to pull things like the waste and the CO2 back into the vessel. Okay, so this one pulls. So now that we know those two types of pressures, let's draw out a capillary bed. I know that my drawing skills leave a lot to be desired, but hopefully it gets the point across. All right, so I'm drawing out a capillary bed, and over here is the arterial end, right? So the blood is coming from the arteriole and into that capillary bed. And at the other end, the blood is going to be draining the capillary bed and moving through the venial. Okay. So the first thing I want to talk about is hydrostatic pressure. Now remember I told you that hydrostatic pressure is kind of like blood pressure. So it's the pressure that's inside the vessel. Now one of the things we talked about early in chapter 19 is how blood is actually moving through your body. It's following a pressure gradient. So blood is always going to move from a high to a low pressure. And in terms of your body and circulation, if you think about it, blood pressure is the highest right where it leaves the heart at the aorta. It's 120 millimeters of mercury at that point. As your blood moves through your arteries, because of friction, it's going to lose a lot of pressure, and it loses pressure pretty quickly. So it goes from 120 millimeters of mercury, right where it's leaving the heart, down to 35 millimeters of mercury by the time it enters that capillary bed. So that's a huge drop in pressure. Okay, so from the aorta at 120 millimeters of mercury down to 35 millimeters of mercury by the time we get to the arterial end of a capillary bed. Now as it moves through the capillary bed, it's going to continue to lose pressure. By the time it gets to the venial end, the hydrostatic pressure is down to 17 millimeters of mercury. And from there, that blood is going to drain into the venial. Those venules are going to merge to form veins, and those are going to dump back into that right atrium. And when the blood dumps back into the right atrium, the pressure is back down to 0. So truly, blood is moving through the body because of a pressure gradient from high to low. It starts at 120, and the pressure continues to drop until you get back to 0. Okay, and then we start all the way back over again as it leaves the heart again. So the numbers that I've written up here, these are our hydrostatic pressures. Okay, now I told you just a second ago that we also have osmotic pressure. So I'm going to draw that in green. Osmotic pressure is tissue pressure. So it's not what's in the vessel, it's actually what's in the tissue. And osmotic pressure is always 26 millimeters of mercury. And it's 26 millimeters of mercury in all of that tissue. So it doesn't change between the arterial end and the venial end because we're not looking inside the vessel. Instead, we're looking in the tissue. Okay. Now, if we look at this, remember, if we just think about pressure gradients, anytime a pressure is higher, blood is going to go from a high to a low pressure. So if we just look at it from that aspect, we can see that the pressure at the arterial end at 35 millimeters of mercury is higher than what's in the tissue. So blood is going to go from high to low. Blood is going to move out of that capillary. And when I say blood, it's not really blood that's coming out. It's going to be things like oxygen and nutrients. If we look at the venial end, remember that hydrostatic pressure at the venial end is dropped down to 17. So it's no longer higher than what's in the tissue. So again, blood is always going to go from high to low. So it's going to go from where it's higher in the tissue at 26 to where it's lower in the vessel. And again, I'm saying blood, but in reality, it's going to be things like CO2 and waste. So this is how, at the arterial end, we can move things out of our vessels and into our tissues, and how at the venial end, we can move things from the tissue back into our bloodstream, okay? And so this is sort of an overview of all of that, but let's talk a little bit about the equation that I put up there for you, right? So the net filtration pressure. In your textbook, the net filtration pressure is a pretty difficult equation, but we can definitely simplify that compared to the way it is in the book, because a lot of those values are negligible that are in that textbook equation. So I've simplified it to just say net filtration pressure is equal to your hydrostatic pressure. Again, this is what's in the vessel. This is like your blood pressure, minus osmotic pressure. And this is what's in the tissue. Okay. Now, anytime net filtration pressure is positive, fluid is going to move out of the capillary. Anytime net filtration pressure is negative, fluid is going to move into the capillary. So let's go back to that picture, and we already know what way the fluid's moving just by looking at it, but let's go ahead and make sure that our net filtration pressure is positive where we think it should be positive and negative where we think it should be negative, okay? All right, so let's do our equation up here. Net filtration pressure is equal to hydrostatic pressure minus osmotic pressure, okay? So we're going to start here at the arterial end. So NFP, net filtration pressure, the hydrostatic pressure is 35, minus the osmotic pressure, which is 26. And we know that 35 minus 26 is 9. That is a positive number. So therefore, we know that fluid is moving out of the capillary. Okay, so we did get that one right. Let's make sure that at the venial end, it also comes out correct. So we can do NFP equals, at the venial end, remember hydrostatic pressure is down to 17 and osmotic pressure is now 26. So 17 minus 26 is going to give us a negative 9. So now we have a negative number, which means fluid's moving into that capillary. Okay, so we did not only just being able to look at this, we can tell going from high to low, we can easily tell what way fluid's moving. But on an exam, I might give you numbers for hydrostatic pressure and osmotic pressure and ask you what is the net filtration pressure. So it is important that you do know how to do those equations. You also need to make sure that you know if it's positive, it's moving out of the capillary. If it's negative, it's moving into the capillary. Okay. So the very last two slides I want to show you, it's the same thing that's in your book, but I like to put it in here because that way when you're looking at your book, you kind of know what you're looking at. So in the equation that I give you, I actually take out these numbers down here because they're negligible. So I'm really just giving you the major numbers here, which is your hydrostatic pressure of the capillary and that osmotic pressure. And so you can see this is at the arterial end, right? So it says up here, use a different color so we can see better. Arterial end, you can tell because it's red, so it's oxygenated blood. But you can see here that the hydrostatic pressure in the capillary is 35 and the osmotic pressure that's out in the tissue is 26. And again, our NFP equals hydrostatic pressure minus osmotic pressure. And again, in this case, the answer is a positive number. So that means that fluid is moving out of that capillary. And in your book, this shows the venule end as well. So you can see up here it says the venous end of the capillary. You can tell, I'll do it in a different color, you can tell because it's showing blue blood or spent blood, blood without any oxygen. And again, we do the same thing. We can see the hydrostatic pressure is, the pressure in the capillary is dropped down to 17. Osmotic pressure doesn't change, it's still 26. So we can do our equation, NFP equals hydrostatic pressure minus osmotic pressure. And this is going to give us a negative number, which indicates that fluid is moving back into the capillary. Okay. Now you'll notice in the equations on both this slide and the next one, here are the equations from your book, which you can see that they are much more complicated. There's more numbers involved. And even though it's not exact, what we get up here is very close to what you get when you add some of those negligible numbers in. It's only off by 1. And the same thing here. You can see that this equation is definitely a little more complicated. And again, we're only off by 1. Okay. All right, so I hope that this helps so that you can actually see the drawing, and I've broken it down a little bit for you. Please let me know if you have questions or if there's any part of this that you just don't quite understand, and I can try to come up with a different way to explain it for you.